Okay, question 3. This is an experimental setup question. So one of those questions that's talking about an experiment, understanding what an experiment is about, and then answering some questions about the experiment. Now, there's a lot of writing here. I'm going to read this. If you want to skip ahead to just us talking about the question, you can skip ahead to that point, maybe about 20, 30 seconds. Succinate is converted to fumarate by dehydrogenase enzymes as shown. So you've got succinate dehydrogenase, fumarate plus hydrogen. The rate of this reaction can be measured using the indicator DCPIP, which changes color from blue to colorless when it combines with hydrogen. An investigation was carried out into the effect of an inhibitor on the rate of this reaction at different concentrations of succinate. 5 test tubes were set up as shown in the diagram. Each test tube contained a different concentration of succinate. So, really important when you do these questions to try and understand what's going on with this kind of experimental setup. Basically, you've got this reaction that takes place, and you've got this indicator DCPIP. When this reaction is complete, so when you've changed this compound into this compound, which has hydrogen, it's going to, your indicator DCPIP is going to change color. It's going to go from blue to colorless. So the more this reaction is going on, the faster you're going to go from blue to colorless. That's what the indicator is supposed to do for us. So they're setting out an investigation which is studying the effect of an inhibitor on the rate of the reaction. So the idea is, if we've got an inhibitor, that should slow down the rate of the reaction, which means it should take longer to decolorize our DCPIP. It should take longer for our test tube to go from blue to colorless because we're inhibiting the enzyme a little bit. Now, the first part of this question, we get a little bit of information about the results, which is important, and we'll talk about that later. But the first question you've been given is to state a variable other than the ones being mentioned that would need to be controlled for a valid conclusion to be drawn. So this is kind of about control, which variables you control and all that kind of stuff. Now, whenever you do any of these questions, you should think of 5 things that need to be controlled in an experiment. Not up to 5, but these are 5 things to always think about when you're thinking of what variables that haven't been mentioned should I mention to get my mark. Always ask yourself, have they mentioned a temperature? Have they mentioned a pH? Have they mentioned a volume? Have they mentioned a mass? Have they mentioned a concentration? So temperature, pH, volume, concentration, mass. If you remember those 5 things, you'll find answering that control variable question super easy. So if we look at this question, they've told us about the volume of succinate, which is our substrate. They've told us about the volume of our inhibitor. They've told us about how much DCPIP they're trying to put in, 1 drop. And they've told us about the volume of the succinate dehydrogenase. They haven't told us about temperature. They haven't told us about pH. However, they will need to be controlled in order to have a valid experiment. So for this mark, we could say control the temperature, control the pH. They also haven't mentioned the concentration of any of those compounds. They've told us the volumes, but they haven't told us the concentrations. So we could say the concentrations of any of those compounds. Okay, mass is not applicable here because we don't have anything solid. If we had a solid piece of something, mass would be important to control. And like we said, we've already discussed volume. So what's it going to look like to answer this question? Well, the answer here is either going to be temperature, pH, the concentrations of the DCPIP, concentration of the enzyme solution, concentration of the inhibitor solution. Any one of those would get you the mark. Next question, describe one possible source of error when using DCPIP in this investigation. Well, in our setup, we said we were going to use 1 drop of DCPIP. So, and imagine somebody's going to be sitting with a dropper, dropping in drops of DCPIP. So there are issues with this because we're entering in some human error. Number 1, you mean to put in 1 drop, but you put in multiple drops. Number 2, how do you know that all drops are the same size? And number 3, you're putting a drop into an experiment. What happens if the reaction's already started taking place before you put that drop in? So these are all things that could go wrong when you're using a drop or using a dropper in an experiment like this, in this case DCPIP. So again, for this mark, you could say it's difficult to measure the end point of the reaction. Okay, that's because you're using a dropper to decolorize something. Again, you're looking at a reaction and you're looking to see the reaction's ended by a color change, and that's completely something that you're judging by human error. Number 2, it's difficult to ensure that all the drops are going to be the same size or the same volume. And the third thing you could say is that you could accidentally add DCPIP after the reaction's actually started to make it so it's not really as valid as it could have been otherwise. So the next question is asking, suggest how the reliability of the results in this investigation could be improved. Now from National 5, there's a phrase I had seared into my mind, repeat for more reliable results. And that is true. But higher, you need to say a bit more than just repeat improve reliability. So you could say two things. You can repeat the experiment at every concentration level, okay? Or you can repeat the experiment and you can calculate an average. And in this question, the concentration level is the succinate concentration, so our substrate concentration level. If you watched one of our earlier videos, we talked about whenever you're looking at an enzyme question, make sure you can identify what's your enzyme, what's your substrate, what's your inhibitor, if you have one, and what's your product. So the succinate is going to be what we're studying. We're changing the concentration of succinate. So we want to repeat the experiment at every succinate concentration, which is basically like doing the experiment again. Or we can do the whole experiment again and calculate an average. Okay, on to the next question. We're being asked to draw a line graph to show the results of the investigation with an inhibitor. So the table you've been given shows you two sets of results, one with an inhibitor at every succinate concentration and one without an inhibitor. And what you're looking at is the time to decolorize DCPIP. So what you're interested in here is you're looking at with inhibitor. So you're only plotting these bits onto your line graph, okay? So the keys to a good line graph are things like scale and accuracy of your points, labeling of your axes and things. Let's look at one that I did earlier, and we'll talk about that one there. But the things that we had to do here to make an accurate line graph is, number one, we had to label our axes. So we had concentration of succinate in moles here. That's taken verbatim from the table. And we have the time to decolorize DCPIP with inhibitor in seconds, again, taken directly from the table. We have a scale for our y-axis going up in tens, okay, and that's talking about the seconds. So from 10, 20, 30, all the way up to 100. And then we have along the x-axis, we have the concentration of succinate. Now, I've decided to do 0, 0.2, 0.4, 0.6, 0.8, and 1.0. And it's using the entire bottom part of the x-axis. The reason is that with line graphs, if you don't use more than half of the graph paper, you will lose the mark for scale and axes. Okay, so we always have to make sure you're using as much of the line paper as possible with an accurate scale. And this just gives us enough space to draw these points nicely. Goes without saying, try and make sure your points are all plotted in the correct place, just so that you're totally happy with where the points are plotted, and then draw a connecting line all the way through it. And that's what you'd have to do to get the mark there. The 2 marks there. There's always 2 marks for a graph question. C, describe how the results show the inhibitor used is a competitive inhibitor of succinate dehydrogenase. Now, to answer this question, we're going to go back to the table. Even though we drew a line graph, let's look at the table because it's a lot easier to interpret what's going on. So we're being told the inhibitor that we used here was competitive inhibitor. How do we know that? Let's take a look. So if we look at our results, we can see that our values for the time taken to decolorize our indicator is different with an inhibitor and without an inhibitor. What you'd expect, because an inhibitor is going to slow the rate of the reaction. So with an inhibitor, we're taking 94 seconds. Without the inhibitor, we're taking 72 seconds at 0.2 succinate concentration. If we focus on without inhibitor for a second, we just look over here, we can see that the fastest we get to is 8 seconds to decolorize the DCPIP. So that's kind of what we could say is the maximum rate of the reaction. That's as fast as the reaction is going to go. It's going to take 8 seconds. And that happens at 0.8 succinate concentration and at 1.0 succinate concentration as well. Now, if you'll notice, with an inhibitor, we still reach that maximum speed. We still reach 8 seconds to decolorize DCPIP. Now, because we've reached the same max value for the rate of the reaction, this has got to be a competitive inhibitor. Let's look at a graph to explain this a bit further. So, if we look at this graph's talking about what happens to enzyme activity when you've got an uninhibited enzyme and enzymes that have a competitive or non-competitive inhibitor. So the purple line is the normal, no inhibitor rate of reaction. The green line, you can see, is a bit slower, a bit below the purple line. Okay, so along here is substrate concentration, up here is rate of reaction. So as we increase substrate concentration, we want to increase rate of reaction. So the green line is the rate of reaction at that substrate concentration is lower than a normal enzyme. However, we do still reach the same maximum rate of reaction. We will still get to the top there. However, with a non-competitive inhibitor, if we increase substrate concentration, still won't increase the rate of the reaction to the same point as a normal enzyme. So if we reach the maximum rate of reaction with an inhibitor, it's got to be a competitive inhibitor, because the only type of inhibitor that can still reach that max rate has to be competitive. Non-competitive inhibitors won't even reach that max rate. So we can say a number of things to basically express this answer. We can say the maximum rate of reaction is reached. That's one thing we can say. Another thing we can say, if we go back to our answer, is that as we increase succinate concentration, the rate of reaction will also increase in speed. Or we can say the time taken to decolorize the DCPIP decreases. So again, once you increase succinate concentration, time taken to decolorize DCPIP decreases. And that's kind of, again, pulling on our understanding of this graph, that if you increase substrate concentration, you will eventually, for a competitive inhibitor, reach that maximum rate of reaction. Whereas for a non-competitive inhibitor, you would never reach that rate of reaction. You could also say that if you increase substrate concentration, you overcome the competitive inhibition. Either of those would get you the mark. So continuing on, succinate is a metabolite in the citric acid cycle. State the exact location of the citric acid cycle in a cell. So this one is one that we just need to know the locations of different processes in the cell. Citric acid cycle will take place in the mitochondria, but it's not enough to just say mitochondria. We've got to say it happens in the matrix of the mitochondria. Okay. So second question, describe the role of dehydrogenase enzymes in the citric acid cycle. Now, dehydrogenase enzymes, people don't tend to like them very much. Let's take a second to remind ourselves of what they do. Don't panic. Every time that somebody looks at this image, they're like, oh my gosh, it's so hard. It is a complicated image. Let's break it down. What we're seeing here is we're seeing pyruvate, which is going to come from step 1 of respiration, glycolysis, and that's going to turn into acetyl coenzyme A. We're going to lose some carbon dioxide, and we're going to take out a hydrogen, which is going to turn NAD into NADH. Then that acetyl coenzyme A is going to join with another compound, oxaloacetate, and become citrate. And round around this circle it will go. It will keep going round this cycle. As it's going round, it'll give off some carbon dioxide, and it will also give off some hydrogen. So what does dehydrogenase do? Every single time here you see that NAD turning into NADH, what's happening is these dehydrogenase enzymes are at work. Their job, dehydrogenase, is to take hydrogen ions and their electrons from the intermediate compounds in the citric acid cycle. I'm going to say that again. Dehydrogenase enzyme's job is to take away the hydrogen ions and their electrons from the intermediate compounds in the citric acid cycle. You also see dehydrogenase in glycolysis, and they do the same thing. They're removing hydrogen and their electrons, so the hydrogen ion and its electron, from a specific compound to turn NAD, which is our hydrogen carrier, into NADH. So that's what you see happening here. All the time you see NADH, what's happening is dehydrogenase enzymes are at work. So if we wanted to put that in an answer, we could say that the role of dehydrogenase enzymes in the citric acid cycle is to remove the hydrogen ions and their electrons. Or we could say it's to pass these hydrogen ions and their electrons to NAD+, so that they can become NADH, basically so they can carry that hydrogen. And those compounds are important for us in the electron transport chain, which is not part of this question as well. And finally, we saw in that diagram there, the substance that combines with an acetyl group to form citrate, that's going to be oxaloacetate. Okay.